Hello, everyone, and welcome to Pocket Size Bio with Dr. Casco. This video begins a series that I've been wanting to do for quite some time, which is giving a lecture of the chapters on the molecular biology of the cell textbook, having the most recent edition by Bruce Alberts, right? This is a staple in molecular biology for undergraduate students and even graduate level courses that could serve as a reference for anybody doing research, right? So I've wanted to get on this undertaking to cover all the chapters of this book. So this is my video of chapter 1, and I'm going to break down every chapter into particular parts. In this case, chapter 1 will be broken down into 3 parts, right? So let's get to it. All right. So chapter 1 is basically cells, genomes, and the diversity of life, where the textbook gives a broad introduction on what is life, right? And some features of what is characteristic about cells, right? So it's quite interesting how they kind of plot this idea of a cell as being chemical factories, right? You have a lot of molecules that are interacting, they're moving randomly, right? And they essentially take in matter to process this material and manufacture necessary components of the cell and ultimately break down material in kind of a synchrony so they can maintain itself, organisms, in particular the cell, right? In synchrony with the environment, right? That being said, this book, of course, focuses on cells and that idea that cells are the fundamental unit of life, right? And the whole focus of cell biology is that concept of studying the structure and function, right, and overall behavior of cells. Okay. One of the key points they mention is that, and of course, this is a defining concept in biology, is that despite all the diversity that we see in this natural world, in this living world, there are some commonalities, right, inside cells and some similarities even with organisms, right? But um, biology as a whole, right, has this kind of counterpoint between these two aspects, right? That there's a lot of variety between even individual particulars, right? Individual organisms that they might vary in their structure, how they behave, and so forth. But there are also some vast similarities, right? There's some constancy in the fundamental mechanisms that govern the behavior of cells, right? That we all have, right? As living organisms. Having that said, the book nicely encompasses some what they mention as universal features of life on Earth when focusing on cells, right? Now, this is a little bit of different on the characteristic of life in terms of like homeostasis and response to stimuli and things like this. It particularly focuses on the intricate mechanisms within a cell, right? And why the cell is that fundamental unit of life. Okay, it does encompass it in a bit. So let's get to it, right? There's several features, approximately 8, right, that the book discusses, right? And one of the things that it gets into is that, um, you know, it reiterates this idea of species, right? And there's so much diversity. There's plus 2 million described species, right, that are unique in their own way. But nevertheless, despite this explosion of diversity, we all are encompassed or revolve around, sorry, this idea that we're able to reproduce and we're able to pass down features that we have, within ourselves onto offspring, right? A concept we all are familiar, which is heredity, right? Passing down our genetic information from parent to offspring, right? Via different methods of reproduction, right? But we have that goal. And in that sense, you know, that's one of the defining features of life, being able to reproduce, pass down information so future generations can also contain the information to thrive and live and produce proteins and things like that, right? Um, One of the things that's, again, central to the definition of life versus things that are non-living is that idea of using free energy. And there's a simple, nice example that the book gives, um, you know, wood burning, a fire, that is consuming wood. It is taking up energy, right? But it's not using that energy like how living systems do to basically um, create and multiply, right? So in that sense, we have this idea of use of free energy from the environment to keep our generations from happening, right? Sorry, from to occur, right? Produce more generations of individuals, more and more and more, right? And one of the interesting facts about cell biology, of course, is that most living organisms, right, are either single cell, or if you're multicellular, ultimately you have to have come from a single cell, right? So, very nicely put in the text, the single cell therefore contains all the hereditary information that define a species, even if you're multicellular, right? The single cell is enough to define that species. So that was a very interesting sentence to read, right? Um, and again, that same idea, right? You within each individual cell, being multicellular or being single cell, you have the necessary machinery to gather raw materials from the environment, process these raw materials, break down these molecules in catabolic reactions, process them, make more complex biomolecules, and use it enough that you can create material enough for you to make a copy of yourself, right? To reproduce, right? In the case of asexual reproduction, just to give you a sense there, right? And if you take a look at this little panel here, again, this is an example of how even multicellular organisms, despite being multicellular, ultimately come from a single cell, right? You have these fertilized eggs of these different organisms here. You have a sea urchin, right? You have a mouse. You have a seaweed, a fucus, right? Seaweed. All of them come from this single cell. Of course, through several cell divisions, they'll end up being into these individual organisms that we think about commonly, right? But we all come from single cells. Okay. Now, talking more specifically about the universal features of life, right? Let's take a look at um, these individual ones, right? So universal feature that I've basically enumerated. The book doesn't enumerate them, but I have enumerated them for you. So universal feature one, all cells store their hereditary information in the form of double-stranded DNA molecules. Okay. So there's an analogy that the book gives that, of course, similar to computers, right? That we store information as bytes, right? And uh, living systems, we can also store information in some physical form, right? Which is DNA. Just like computers can, let's say a USB drive or a floppy disk or, disk in general. Living systems have their own form to store information, which we know as DNA, right? So all living things, on Earth have inherited the form to have their genetic information in some physical way, right? Which is DNA, which means that we, in a sense of evolution, we must have came about from a, a universal common ancestral cell that had that initial machinery to be able to store information, right? Which probably came about around 3.5 to 3.8 billion years ago, right? And it's quite astonishing. It is um, mind blowing, if I can say, how that idea of passing information has been, it arose and was preserved and is still coming on. Of course, with some alterations and modifications and enhancements. Um, that nucleic acids and DNA has been the go-to biomolecule to do this, right? And it's been so well um, in doing its job that it continues till today, and who knows for many, many years to come, right? So, yeah, this DNA, it forms information, stores information, right? And basically, in the form of a double-stranded molecule of DNA, right? And some general properties that we should, of course, we all are aware of, that we know of. But just to kind of reiterate here, a DNA is a double-stranded nucleic acid, macromolecule, polymer, you can also call it, right? That is very long, unbranched, right? And basically they're paired polymer chains. There's two nucleic acids that are basically bound together, right, by hydrogen bondings on the nitrogenous bases that we're going to take a look at, right? What's interesting is that, these long polymer chains are composed of four repeating monomers, right, that we commonly know as nucleotides, right? And there's four types of nucleotides: um, adenine, thymine, guanine, cytosine, right? And these monomers, they repeat in different sequences and different combinations, but ultimately DNA is composed of just these four molecules, right? And you see that in all forms of life, in every organism, right? So there's a consistency with life and this ability to store information, right? And what is a very interesting, that there's such a consistency in life that sequences can be inserted from one species onto another species. Okay, of course, with some little modification and some little processing, general little effort nowadays in molecular biology laboratories, right? You can basically get a gene from a human and insert it into a bacteria to produce that particular protein. That's how consistent DNA is, right? Um, between different species, right? Ultimately, information can be successfully inserted, read, interpreted, and copied, right? So that's that in itself is quite astonishing, right? So, um, of course, and by understanding that throughout the history of scientific discoveries, scientists can now read and sequence the complete DNA sequence of many species, basically sequencing the genome, right, of many species. But what's even more so recently with single cell sequencing has created even more powerful tools to understand and analyze, right, DNA, right? Um, we all are familiar with the genome, right? The totality of hereditary information, right, that an organism can pass down from parent to offspring, right? So, number one, right? All cells store their hereditary information in DNA. Going into more specific, um, going into more detail regarding information, right? You have universal feature number two. All cells replicate their hereditary information by templated polymerization, right? Templated polymerization. So, um, you know, life is possible again just because of the nature of the structure of DNA, right? It's double stranded. Due to its structure, it is able to store information, process that information, you know, and extract, be able to produce a protein from that information, right? And this undergoes several steps, right? In this chapter, it very briefly talks about it. In the later chapters of this textbook, it goes into extensive detail, right? But this is essentially transcription and translation, right? and DNA replication. So all of this is used by DNA um, templated polymerization, right? And the first thing we're going to talk about is DNA replication, right? So just some little kind of notes here that are maybe familiar to you. Nucleotides, as we know, the little building block, the monomers of DNA, right? They consist of a sugar, right? Of a 5 carbon sugar called deoxyribose, right? That is attached to a phosphate group or has a phosphate group attached to it, right? And also a nitrogenous base. Okay. And I'm going to just show you here. So we can reference on the little figures here. This is basically a nucleotide where you have your sugar and you have your phosphate that is bound also to a nitrogenous base. Okay. And in this little depiction here, you have this little kind of full nucleotide. All nucleotides have that sugar phosphate um, um, consistent section, right? And a nucleotide could vary by the nitrogenous base, which you have the 4 of them, right? So you basically can connect nucleotides on the phosphate, on the sugar phosphate um, molecules that creates a chain, right? And the nitrogenous bases are nicely pointed, pretty much in a 90-degree angle, if I can say, right? To create a nucleic acid strand, right? So that in itself is a, DNA strand, but this is single stranded here, right? What happens then is that you can get 2 nucleic acid strands of DNA and they basically bond together, right? Um, where nucleotide base pairs, right? The nitrogenous bases are complementary to each other. And by what is called templated polymerization, you can get new nucleotides coming in and you can get a newly synthesized nucleic acid strand of DNA to come in using a template, right, of DNA. These nucleotides could come in based on complementary base pairing rules, right? They can come in and bind where you have G always binding to C, okay? C always binding to G, A to T, T to A, and so forth and so forth. And they always connect in a series, basically, right? As you can see in this picture here, G is coming in and it's attaching. And what's quite consistent is that sugar phosphate that basically like Legos just comes in and nicely bonds, covalently bonds from one to another and one to another. But what you do get from one nitrogenous base to another, you get a hydrogen bond, which is a more temporary bond, right? As we know, right? So ultimately, you get this chain together, and as a result of the hydrogen bonding from these new nitrogenous bases, you know, you create this double helix conformation, right? Um, Ultimately creating that DNA double helix, right? So in general, replication, whenever DNA wants to um, copy itself, right, it uses each strand of the DNA as a template, and then free nucleotides that are floating around, right, can come in and just attach in a particular sequence, right, that we'll get into in the later chapters. Okay. Um, Living cells cannot synthesize a free strand. So you have to have a template, right? Generally, of course, having this complementary rule pairing, A to C, G to C, A to T, and G to C. Sorry. Okay. So this whole process of just copying DNA and duplicating DNA is referred to as DNA replication, right? And essentially, you know, no matter which organism, eukaryotic cells, even prokaryotic cells, bacterial cells, mammalian cells, reptilian, cells of different, all the variety of life, the process is universal. You're using DNA as a template, as a method to copy DNA, right, and produce more DNA. Okay. Just to give you guys here an example of um, an overview of the visual there. Okay. Also, here is um, a visual, right? A diagram showing this copying of DNA, essentially referred to as a semi-conservative model, which we'll see later. Right? We have a DNA double helix, basically unzips. Each strand that you see in dark red serves as a template, and you get new free nucleotides basically coming in and joining to each strand, and you have two new strands of DNA. Okay. Okay. Now we're going to look at universal feature number 3. All cells transcribe portions of their DNA into RNA molecules, right? So DNA, of course, we've seen how, or gotten a sense of how it can multiply and replicate itself, right? But with that information that it carries, how does it get expressed? How does it get used, right? So DNA needs to express the information it carries, right? And this is done so by producing two other molecules that are very important in biology, right? Needed for all life, which is essentially RNA and protein molecules, right? And the whole general process in which this occurs, right, where you're expressing the information of DNA is done so through a couple of steps, right? So we've talked about DNA synthesis and replication, but having that DNA strand and having a particular section of DNA, basically the gene, you have something called transcription, which is RNA synthesis, and then using RNA as a template, you have protein synthesis, which is called translation. Ultimately, you're producing a protein from information that is carried on the DNA, right? So, um, more specifically, right? This whole process of expression, of DNA expression, it begins with templated polymerization, right? Um, Called transcription, right? You use DNA as a directed template to synthesize RNA, another type of nucleic acid, right? Single-stranded, that is referred to as ribonucleic acid or RNA, right? And ultimately to produce a particular type of RNA that is called mRNA or messenger RNA. And something about RNA is that it's being a nucleic acid, it uses the nucleotides with the exception of one nucleotide that it replaces, which is thymine. It replaces it with uracil, right? So it uses a different nucleotide, right? Very similar to thymine, okay? And all the others are the same. A, G, and C are still used, right? So the outcome of this process of transcription is basically a single-stranded polymer, right? That represents a particular section of that DNA, right? A particular gene of interest that needs to be activated at this particular time for whatever X reason, right? So, um, interesting that that RNA that was synthesized, could be used multiple times. It could be read by ribosomes multiple times to produce that particular amino acid strand, right? For example. So RNA in that sense could be mass produced. And then when it's not needed anymore and when that protein is no longer needed, it could be disposed of and degraded and so forth, right? So that's transcription in a nutshell, right? You're producing an RNA sequence coding for a particular gene um, from a DNA template. And then you have translation where you're using that RNA strand to essentially be read by a ribosome to produce an amino acid strand. So you're even going from a different language of nucleic acid to amino acid um, molecules, right? So translation is referred to as being mRNA directed, right? You're using mRNA as a template, and you're producing an amino acid chain ultimately to produce a protein, right? In a nutshell, that is how a gene gets expressed, right? Universal feature number four, oh, of course, always going relating to this, pertaining to the previous one, all cells use proteins as a catalyst, right? So ultimately, why do we need to express genes is to create a protein that we're going to use one way or another, right? Um, and proteins, right? Um, they're similar to DNA and RNA in the sense that they're generally long and unbranched polymer chains. So they're just a single strand, right, of amino acids, right? The building blocks for proteins are amino acids, right? We're going to take a look at in the later chapters. Okay. So there's a standard repertoire of um, amino acids, right? Uh, that essentially naturally occurring are around 20 types of amino acids. Okay. And depending on the sequence of RNA, right, you will correspond to a particular amino acid. Okay. And these amino acids get strung together in a sequence. And ultimately, that defining sequence and that order will create a particular protein. Okay. And different amino acids, this is beyond the scope of the chapter, right? Have different properties, but depending on the different properties, for example, some amino acids are polar or they have polar side chains, some of them are non-polar, right? Um, uh, you will create a particular protein, okay? And by having a specific sequence of amino acids, you create a specific protein. Having a specific protein with a predefined amino acid sequence, you create a particular structure. And as we know in biology, the structure fits the function, right? So a protein having a specific structure will ultimately define its function, right? So again, when we get into protein structure, you'll see this in more detail, but the sequence of an amino acid will determine how it will fold, right? Creating that primary structure, secondary and tertiary that we're familiar of, right? Um, ultimately giving that three-dimensional structure of the protein. Okay. And through time, of course, you know, several proteins that have been useful will ultimately stay within the organism to be providing that particular function, um, enhancing the rate of a reaction, breaking down certain molecules or synthesizing molecules better and better. So, um, ultimately, those proteins that are beneficial will essentially stay with the organism, right? So, um, in a broader sense, proteins, they are able to carry out, depending on its structure, many, many functions in the body, many, many functions in the cell, right? From forming structures, maintaining structures, regulation um of gene, right, activation or inactivation, um, movement, sending signals, etc. There's such a myriad of functions that proteins can do, but for now, just know that they do have many, many functions, right? And of course, how do proteins work is because they have specific areas where they bind to other molecules, right? They have reactive sites. Let's say in the case of enzymes, where they bind to a substrate, right? To essentially catalyze a reaction, right? Ultimately, by breaking or creating bonds, they help enhance a reaction, right? Or speed up a reaction. Okay. Um, what is interesting to note that the book very nicely puts in a sentence, proteins are the main molecules putting the cell's genetic information into action, right? It is the result of the DNA's information being translated into protein that basically shows it, if I can say, as the effector, right? um, from the cell's genetic information. Okay. And in regards, talking a little bit more about catalysts and living cells, catalysts, right? They speed up specific chemical reactions, incredibly specific chemical reactions, without themselves being changed, right? Basically, in essence, being an enzyme, right? And if we were to think about how a living cell actually functions, living cells are essentially self-replicating collections of catalysts. You know, you have this big um, uh, combination of chemicals within a little sack of more chemicals, basically the cell membrane, and they're just reacting together. Things are occurring, things are being made, things are being broken. So a cell is basically a self-replicating collection of catalysts, right? Cells take in food, process it, right, to provide the building blocks to make bigger, more complex molecules, right? Provide energy to other uh, um, signaling pathways, for example, right? And at the same time, once these reactions have occurred, basically the waste material needs to get discarded and so forth, right? In that sense, you have kind of feedback loops that connect proteins, right, and polynucleotides that are referred to as kind of being autocatalytic, right? Self-reproducing, showing self-reproducing behavior within cells, right, of all living organisms. If you take a look here, right, this can get a better depiction of what I'm trying to say here. Um, in general, a cell, right, food comes in and takes food in, right? That food is broken down, and then you have those building blocks, right? As it's also being broken down, it also provides some energy, right? These building blocks in turn will serve as the monomers to create proteins or more complex um, structures that could be used for the cells, a collection of catalysts, right? Where it could be used in many cellular processes. Okay. Anything that isn't used within the cell ultimately will get discarded. Okay. And then you have here where you have this kind of autocatalytic cycle, right? Where you have nucleotides that also come in, right? If you remember from DNA and RNA also, right? You have these little building blocks. It synthesizes DNA and RNA. That in turn has the information to create proteins. And then that information is used, it creates the protein, and then the protein itself can serve as a catalyst to create even more DNA and RNA through the process of replication, transcription, where we're going to see in the later chapters, you have DNA polymerase, RNA polymerase, for example, right? And so forth, right? So it's interesting that you have a cycle occurring between these two molecules. Okay. So now we're going to go to universal feature number 5. All cells translate RNA into protein in the same way, right? So in terms of mRNA sequence and protein encoding, right? Information that is in the mRNA sequence is read out, right? It's read out in group of 3 nucleotides, which are commonly called codons, right? Sets of 3 nucleotides. Let's say AUG, UUU, and so CCG, and so forth. And each codon that is on the mRNA template, right, codes for a single amino acid, right, that corresponds to what is called a tRNA, right? And before I get into tRNAs, right, there's a couple of combinations that could be done based on the different nucleotides that we have available, right? U, A, G, and C. So for every set of 3 nucleotides, for every codon and the possibility of nucleotides, you can make 64 possible codons, right, from the 4 nucleotides. But only 20 of them are naturally occurring amino acids, right? So you can have multiple codons that can actually code for the same amino acid, right? And that's actually helpful in terms of mutations that we're going to talk about much later on, right? Um, so and what it actually codes for, each codon, it corresponds to a particular tRNA, right? A tRNA is another type of RNA that is referred to as transfer RNA that comes in And it's a small molecule that on one side of the molecule, it has a small 3 set of nucleotides that binds to the codon referred to as the anticodon, right? And on another side of the molecule, you have one amino acid, right? And so depending on the sequence of mRNA, right, you're going to get a particular tRNA that brings in a specific amino acid, right? And these amino acids, they will start chaining up, right? They'll start binding together to make one sequence of amino acids, right? And that's essentially what translation um, uh, does, right? That's essentially the process of translation, right? And all of that is occurring on the ribosomes, right? And these are big. You can think about them as like multimolecular machines composed of protein and ribosomal RNA that are essentially the manufacturers that create that amino acid strand, right? Sequence, ultimately, which will create a protein. Okay, so that's UF number 5. Feature number 6 is that each protein is encoded by a specific gene, right? So DNA molecules, they are typically large, incredibly large they could be, right? And they can contain many, many, many genes, right? Or sequences of DNA that can code for proteins, right? And RNA molecules, right? And as we get into the later chapters, right? Specific DNA sequences, they're very defined, right? There's a starting point and there's an end point for any protein or RNA molecule that needs to be made, right? So they're very specific in that sense. And these, gene sequences are basically individual segments can get transcribed into separate mRNA molecules, right? That basically will produce that protein. Now, um, for simplicity's sake, right now, each DNA segment represents one gene, right? Which could encode a single protein or some variants, closely related variants. We'll get into that, right? Or even a single RNA molecule. That being said, right, DNA segments could transcribe into non-protein coding RNA molecules, right, that have some other functions, right? They don't get translated into proteins, but they're also counted as genes, right, despite not producing a protein. Okay, so if you see this in highlighted, it well defines what genes are, right? So genes are defined as DNA segments corresponding to proteins or RNA molecules, right? And gene expression in cells is regulated, right? Which kind of adjusts transcription and translation rates depending on, of course, the needs of the cell for a particular thing, for a particular protein, right? So, um, as we're going to see much more in detail later on, right? There's between protein coding segments, there's a lot of regulatory elements. There's a lot of regulatory DNA regions that can control transcription rates, right? And the how it's organized, right, does vary between organisms, between phylums, between different living things, right? But the overall regulatory mechanism is universally the same, right? So, yeah, in regards to the whole cell's genome, right? Um, don't doesn't determine only the nature of its proteins, but also can regulate when it needs to be done and where it needs to be um, created or where that protein is needed, right? So there's a kind of spatial temporal factor that occurs, in terms of gene expression, right? Factor number 7, life requires a continual input of free energy, right? So in essence, we living organisms and individual cells, more specifically, right? Um, you need to be in uh, in constant motion in terms of molecules, in terms of concentration gradients, and ultimately, if a cell reaches equilibrium, chemical equilibrium throughout the entire cell, essentially that's when a cell dies, right? Um, there's no more um, disorder to create um, um, pathways or to create concentration gradients, like I was mentioning before. So, in order to avoid getting into like this ultimate equilibrium, chemical equilibrium, you do need energy, right? You need free energy and you need raw materials to continually grow, reproduce, create material to keep from being in chemical equilibrium, right? And the way that we do that is basically by collecting energy, right? Free energy. And um it's just one of the fundamental factors of life that you need it. Without free energy or collecting energy and consuming energy, basically cells would die, right? So um, it is quintessential, if I can say, for a cell to thrive, right? Essential for genetic information to basically reproduce, pass down from parent to offspring, and so forth, right? Um, and Yeah, more specifically, when you think about cell molecules, right? They could be just random motion of molecules of objects, having a certain amount of energy, of thermal energy, moving in any direction, right? Having collisions going in different directions, and you want to be having that. You want to be having um this type of motion, right, in this molecular landscape. Okay. Um, it's some examples of which we would need this occurring, copying genetic information, DNA sequencing, where you're having random motion of the nucleotides that are floating in the air. They ultimately need to bounce into the template DNA strand, right? To basically make a copy of DNA. Imagine if you didn't have that molecular motion there, um, nothing will get done. So that's what the book is trying to mention, right? You need to have this molecular motion. And then once the nucleotides get bonded, right, you need to have enough strength to resist detaching the nucleotides, right? So um, uh, you need to have strong bonds to resist that disordering effect of thermal motion. So you need to have be able to overcome that random motion, right, of the molecules. Okay. So right, chemical processes in cells require free energy for the creation of order, right? And like I mentioned before, cells need free energy to replicate genetic information with incredible precision, right? And also to synthesize molecules with specific functions. Okay. Um, how do we get free energy to begin off? It's basically that chemical energy you think about, right? Um, Animals' cells derive free energy from chemical bonds of food molecules that we have. Plants, they basically get it from autotrophic processes like photosynthesis. Okay. Factor number 8, universal factor number 8. All cells function as biochemical factories. Now, having this idea of DNA replication and transcription and translation and protein synthesis, right? We kind of start concluding this overall um, structure, entity that can be able to thrive in its environment, right? Um, so ultimately, cells are biochemical factories, right? Cells need DNA and RNA and protein production. And um, ultimately, to do even these things, you need to be able to um, take in material, contain material, manipulate material, raw material, right? More specifically, small organic molecules, right? Um, Sugars, nucleotides, amino acids, moving around, even water, solvents. Um, necessary to be able to thrive, right? So one of the ways to do all these metabolic processes is basically through the use of ATP, right? As we probably are familiar with, ATP is adenosine triphosphate, basically a phosphorylated nucleotide, right? That essentially serves as like chemical batteries for chemical reactions to occur, biological reactions to occur, right? Um, they serve as these energy providers for DNA and RNA synthesis and signaling transduction and many, many molecular processes, right? Um, because they're the carriers of free energy, basically by carrying a third phosphate that is quite energy rich and unstable, if I can say, right? So, in that sense, right, cells function similarly to chemical factories, right? Where you're having some type of transaction of small molecules to create larger things and breaking down things and so forth, right? Um, plants, or photoautotrophs, moreover, use sunlight energy, right? Which use very, very basic materials. You have water and carbon dioxide as the essential input molecules to produce, sugars, right? To produce organic molecules. So, um, one way or another, you're seeing this analogy of biochemical factoring, warehousing, even, right? Other organisms like animals and some bacteria feed on living or once living organisms, right? Of course, being heterotrophs, we need to consume, right? Um, food that has basically pre-made organic molecules, right? It's a little bit difficult for us to make um, organic molecules from nothing, from just sunlight. All right. So, um, factor number 9, right? All cells are enclosed in a plasma membrane, right? Across with across which nutrients and waste materials must pass. So, the cell membrane is one of the most fascinating things I've ever seen studied in biology, and its ability to be very selectively um, choosing what comes in and out is incredibly interesting, right? As you may know, right, living cells are enclosed by a plasma membrane or a cell membrane, right, that acts as a barrier, incredibly selective, that essentially enables cells, right, to concentrate nutrients on one side of the cell, in the extracellular um, matrix, and so forth, right? So there's tight regulation of things coming in and things coming out, right? Um, and without a cell membrane or without a plasma membrane, definitely you cannot have a cell maintaining its integrity, maintaining its shape, maintaining a a balance between material coming in and out, right? And the way that this works is essentially that it's composed of um, phospholipids, right? A phospholipid bilayer, which has a hydrophilic region and a hydrophobic region, right? As you can see in this diagram here, if you were to get these, phospholipids, sorry, let me move down a little bit, these phospholipids um, into a beaker of water and oil, you would have in this little area where you have oil and water, the phospholipids would redirect themselves in such a way that the hydrophilic head will face the water being a polar molecule, and the hydrophobic tail, right, will be facing the nonpolar solution, right, or substance, right? And if you just have phospholipids inside a very polar molecule, solvent, it would actually create a bilayer to avoid the hydrophobic tails from any exposure to this solution, in this case, water, hence creating the um, this basically birth to the plasma membrane, right? And plasma membranes are pretty much referred to as being amphiphilic, right? Consisting of that hydrophilic and hydrophobic parts. My apologies for the video kind of stopping there, but not important. Um, so yeah, you need to have this a bilayer to be able to move material in and out. And the cool thing about the plasma membrane is that some solution, some material can still pass through, um, while others can't. But you know, special proteins are actually embedded within the plasma membrane that can help pass material in and out, right? And that's a whole study in itself, incredibly beautiful. Um, um, a part of biology, right? Protein biology on the cell membrane. So, as I mentioned before, you have specialized proteins that are embedded in the plasma membrane, right? They could be channel proteins, carrier proteins, which help material kind of traffic in and out of the cell. You also can have catalytic proteins that are also in the vicinity of that to carry out functions. You have proteins that can send signals, a signal transduction pathway, right? And so forth, right? So many, many functions can occur from proteins that are embedded in the plasma membrane. So again, same idea, back to this concept about genetic information. DNA sequences have the information to create proteins that will have a specific structure and function to go to the cell membrane and provide a particular function, right? So, um, yeah, all cells are enclosed in a plasma membrane in which nutrients and waste have to pass through. All right, we're almost done with this first part of chapter 1. Universal feature number 10, right? Cells operate at a microscopic cell dominated by random thermal motion. I did mention this before about how you have free energy, and then you have molecular motion, right? Moving throughout the whole cell. Generally, um, you have material moving from high concentration to low concentration, essentially diffusion there, right? But just to start from the top there, right? Cell organization, sorry. Cell organization and internal dynamics, right? The cell definitely is more than just a self-replicating bag of chemicals and macromolecules, right? There's a lot of order, there's a lot of structure there, there's a lot of organelles and individual components that are able to self-replicate. So you do have a lot of organization within the cell, right? Ultimately, to have some synchrony, like an orchestra, to um, uh, make the cell function as efficiently as it can, right? Um, in order for that to happen, you need contents to be, always in motion, right? Moving around. You need some material to be taken from one area to another. You have motor proteins that can move even the entire cell from one place to another, right? Displace the cell. So, and all of this, the majority of this requires energy, requires ATP hydrolysis energy. And some examples of this is cell propulsion, intercellular translocation, or even ion pumping, right? So you need to have these things in order to keep the cell, right, in in synchrony, right? So, more specifically, thermal motion, observed as Brownian motion, is basically influencing diffusion rates, right? You have diffusion that is occurring. Depending on that diffusion rate, you're going to have certain biochemical reactions occurring, of course, depending on concentration gradients. And um, even entire cells, right, moving from one place to another because of this Brownian motion. And when I mean this physical motion of the cell, there's something referred to as the Brownian ratchet phenomenon that I'll get into in a second, right, which can direct a cell, the movement of a cell from these um, protein filaments, right? So essentially what happens, right? You have movement at an incredibly small scale, micrometer scale, due to high viscosity drag forces from water molecules. What happens, and I'm gonna zoom out a little bit, you have this little plasma membrane and you have this little filament here. Um, Due to some resistive forces of water molecules, you basically will get, directed movement by motor proteins will essentially stop. And whenever they stop, you can get some kind of vibration motion in the plasma membrane. And when the plasma membrane basically kind of, if I could say, waddles or kind of waves opposing to the active filament, you can get a small little, uh, monomer of this filament to kind of join in, and then that extends the whole cell shape to go to a forward direction. And then this will keep happening, and then the filaments will extend more and more and more because of that, random thermal motion. Then the membrane will kind of vibrate again, and then you'll have some space. Another little molecule will come in, and so forth, and so forth, and so forth, until you need to go to where you need to go, right? So this is the Brownian ratchet phenomena. One way in which random motion is helping cell propulsion, right? So again, cells operate at a microscopic cell dominated by random thermal motion. Okay. And lastly, um, universal feature 11, a living cell can exist with 500 genes, right? Um, Not too much on this little section here, but in terms of genome information and cell complexity, right? Genomes contain information of all the proteins and all the RNA molecules of a cell, right? And through catalytic reactions, we're able to produce building blocks, right? Um, to enable a cell to thrive. Complexity of living cells is always a topic of interest among scientists, right? What are really the minimum requirements for a living cell? You know, um, they do say complexity can be measured by the total number of genes in an organism's genome, right? Mycoplasma genitalium, a bacteria that has one of the smallest known genomes, has essentially 525 genes, right? Essential genes. Just 580,000 nucleotide pairs, right? Nucleotide base pairs, which is quite interesting. Equivalent to 145,000 bytes of information, which is roughly a chapter's worth. So that's so interesting that you have, with this little amount of data. I don't want to say little, that's an understatement, because that's still quite a lot, 525 essential genes. But this amount to create an organism, to make this organism thrive, is very interesting, right? So the chapter ends with this little sentence here: cell biology, while complex, is not unimaginably so, right? So that is quite interesting, right? Um, so this finishes part 1 of chapter 1 of molecular biology of the cell. I hope it was beneficial for you, and I'll continue making these videos if you guys find it interesting. Okay. Thank you guys for watching, and I'll be back with another one.